And we are really excited to have Dr. Anshu Bhardwaj with us, who would be talking about understanding and addressing genotype-phenotype correlation in complex diseases. As we have been discussing, Dr. Anshu Bhardwaj is a biologist and a data scientist. She has obtained her PhD in 2008 in understanding of genotypic-phenotypic correlation in human mitochondrial diseases from CSIR CCMB Hyderabad. She has over 40 publications to her credit and have trained over 500 young students, and also has served as an associate scientific advisor for science translational medicine, and is on the editorial board of Journal of Genetics and Frontiers of System Biology, Protein Bioinformatics, Computational Genomics, and F1000 Research Galaxy Gateway, where many of our students have already published some of these articles that they have worked on. So we are really delighted to have you, Dr. Anshu Bhardwaj, and we are looking forward to your presentation and learn from your talk. Thank you. Over to you, ma'am.
Thank you so much, Mohit, for that nice introduction. So can you hear me clearly?
Yes.
All right. Are my slides visible?
Yeah, they're coming up. Yeah, they're visible now.
All right. So first of all, thanks for giving me the opportunity to actually talk to students. That's my, that's my lifeline. So what I decided today, since the entire symposium is organized around bioinformatics and data science training and research, I have kept the talk very, very simple, but we have included one component of our work so that you can also see the application of how it can be done, again, in the most simplest possible fashion. So my lab primarily works in two domains. One is basically genotype-phenotype correlations when it comes to infectious diseases as well as mitochondrial diseases. And the second vertical, which is a smaller component, is computational drug discovery. So just a second. So as you can see here, there are several projects that we have done over years. But today I have selected projects which are on the bottom panel of this timeline, which is mostly to do with mitochondrial diseases, because I thought genomics is something which is basically picking up the pace. So it's important that we understand the genomics basics in terms of where are we going, where do we stand today. So as we all know, DNA is the blueprint of life. And one of the first objectives is to actually learn to read this DNA, right? That was the first thing we started doing, because till the time we read what is in the blueprint, we cannot make sense out of it, right? So in early sequencing days in like 1960s, the first RNA sequence, which is of alanine tRNA, was sequenced. It took 5 people for 3 years working to determine just 76 nucleotides. So this is how we started sequencing. We were not sequencing genomes in the beginning. We were trying to standardize the technology to understand how you read this. And subsequently, over years, we started taking baby steps, where we went ahead from that to 1 base per month, then to 1,000 bases per day. And by the time we sequenced the human genome, we were already doing 10 million bases per day, right? Until that time is when, we actually, this is a quiz for kids who would like to enter their answers to say what this event is. So this is basically a landmark event in the history of current biotechnology and data sciences, is when we launched the draft of the Human Genome Project, right? And this is the big machine that at that point was used to sequence it. It took effort from several countries and a very large number of sequencing centers to give us the first draft of the human genome sequence, which was 23 human chromosomes with 3 billion bases, right? Why were we interested in sequencing? Simple reason: to understand human disease and evolution. If you can compare the genome sequence of somebody who is diseased versus somebody who is normal, the anticipation was that we will be able to understand diseases better at a molecular level. And if you can understand something better, it is easier to treat it. So as the previous speaker was referring to a large number of methods for this kind of data analysis, the objective is very clear, that you need to understand human disease and evolution. And that is why there was an intense amount of effort put into place. But this was only a project where you were trying to understand and standardize the technology. And then we went ahead from this basic sequencing methods to something we understand as next generation sequencing methods. And with the advent of NGS, now there are a very large number of sequencing projects going on across the world, where a few countries are actually sequencing their entire population. So these sequencing efforts actually have gone, the scalability is unimaginable. We are generating data at a pace and we don't really know how to analyze it. So that is where the power of data sciences lies in. It is very critical right now for students who are basically deciding on a research objective. Try to learn data analysis, because you actually have a lot of data available that needs to be analyzed in context of what is now disease associated among all this plethora of sequence that you see. And the starting resource that I would recommend to all of you is GenomeAD. So Genome Aggregation Database is trying to aggregate all the genome sequencing projects at one place. So you don't have to wander around several places for this kind of data. So you can go to GenomeAD for that reference. Now, when you say you are trying to compare individuals with and without disease, what are we trying to compare? Or what are we trying to look at? We are trying to come back at a point where we can look at genomic variations, right? Now, genomic variations can be of several types. The most common ones are single nucleotide polymorphism. These are called, in general language, substitutions. We'll read more about them in the next slide. Then there are indels, which is insertion deletions, and they can actually change the protein function. Then there are structural variations. Then there are tandem repeat variations, segmental duplications, translocation. And most of us in our undergrad or in masters must have come across these terminologies, where these actually are impacting the way the chromosome is, or the sequence is arranged in an order. And then when that arrangement goes haywire, you expect to see that there can be a disease phenotype. Now, when you talk of single nucleotide polymorphism, it means that it's a variation at a single nucleotide. Say, for example, let's say there is a gene A, and that gene A has an exon, right? We all know what exons are. In exon 1 at some position, let's say 500, in a population, you either see A or C. Now, in technical terms, we call alleles at the nucleotide level. So there is an A allele and there is a C allele. Now, let's say A is the most common allele, like 90% of the times, A is observed. So we call it as a major allele, and C is observed the rest 10% of the time. So it's a minor allele. So since humans are diploid, we get one pair of chromosomes from our mother, the other from the father. Then, so basically what you actually will see is that either the individuals will have AA, AC, or CC, right, at this particular position. Now, how do you decide that it is an SNP? Is when the minor allele, and in this case that is C, is present at a frequency of more than 1% in the population. So let's take two examples. In the first case, if 94% people have C, which is the major allele, and 6% have T, which is the minor allele, you say this as an SNP. In the other case, if you see a similar situation, but the percentage of the minor allele is not even 1%, that you start calling it a mutation. Now, this you need to understand is heavily context dependent. The reason being that if you are looking at rare variation, then this definition will change. And that is one has to now look at single nucleotide polymorphism definition or genomic variation in context of what you are trying to look for. And obviously, when you are looking for rare variants, you need to sequence a very, very large number of individuals. And then, of course, when both the maternal and the paternal allele are same, that's an homozygous SNP. When they are different, it's an heterozygous SNP. And they are present everywhere. Like they can be in UTRs, introns, splice site. They can be in coding regions where they may or may not change the amino acid. In almost all these cases, they might have an impact on the phenotype. Now, the question that you might ask is that you sequence an individual, sequence another individual. So one person has disease X, the other person does not have disease X. Just compare the sequence and you are good to go, right? You can now actually identify the differences and say this is what is causing the disease. It is not that simple, and that is where what we are discussing today. Any complex disease is not happening because of a single genomic variant. there are many genomic variants which we call as polygenic, in interaction with the environmental factors are actually causing the phenotype. In this case, I have shown the example of coronary artery disease. So complex disorders, I must repeat this because this is a very, very important factor, results from contributions of multiple genomic variants and genes, and that is why they are called as polygenic, and they work in conjunction with the physical and the social environment to bring out the phenotype. And this is the challenge we are trying to now resolve. When you compare two individuals, you will get anywhere between 3 to 5 million SNPs which are different. Which of those SNPs are associated with the disease? That's the first question. Second, which of them might cause the disease? There can be several reasons why the disease is manifesting. And that is where the challenge lies. In order to address this challenge, there are two basic approaches that are applied. One, we call it as a candidate gene approach. The other one is a genome-wide association studies. And we'll very quickly look at both of them. What do you mean by candidate gene approach? So in a candidate gene approach, what you actually are doing, you are basically making an intelligent guess. Like say, for example, you are interested to identify a genetic risk factor for a particular disease condition, which is a complex disease. And you now need to know what are the genes contributing to this phenotype so that you can now look at the variation in those genes. Now, this mostly is done within family members, or it can also be unrelated cases. But the point is you need to have some estimate of what kind of biological components might be involved. Say, for example, I've taken an example here. If you are studying alcoholism, why some people are getting addicted to alcohol, and you want to know the genetic components of that, the most logical choices would be to look at their alcohol metabolic pathways. And particular genes like alcohol dehydrogenase or aldehyde dehydrogenase, these are logical options for you to start. And if you do see that there are several alleles of these genes in a population, making it a very strong case that you now know that they are actually vary among individuals, now you can actually apply a candidate gene approach, start looking for variation only in these set of genes, and then identify polymorphism which might be associated to the disease, and subsequently establishing why this might also cause the disease. So here you are actually also digging deep into the mechanism of what that variation might be doing. The problem is, in most cases, this kind of educated guess either has failed or it is extremely difficult to make. And that is where genome-wide association studies are done, where you are actually following a very data-driven approach. You take two cohorts, two groups of individuals, one normal for the phenotype, the other one actually has the phenotype. And when I say phenotype, I'm saying the disease. And then you sequence them, compare, identify SNPs, and then start looking for association of these SNPs with the disease phenotype. Now, the word associated is kept very, very categorically here. Reason is association is not equal to causation. You may find SNPs which are associated with the disease. They might not be causing the disease as such. So GWAS actually will help you identify, at least shortlist those candidates and then explore them deeper with respect to what is their role in disease manifestation. Several studies have, actually there are many GWAS that have been done till date, and we do have some success stories where if you look at this publication, they actually have listed several traits against which genes were identified using GWAS, and they ultimately actually allowed discovery of drug candidates to treat these diseases. So we do have success stories, means these methods work. Obviously, the number of GWAS that has been performed versus the outcome is not comparable. So we need more intelligent methods of doing that and maybe a better readout. So what actually is going on right now is there is a very big effort going on to catalog genomic variation of different populations all across the world, and then also trying to assess which of these genomic variation might have a clinical implication. So there is, if you look at, I have listed a few papers from last 2 to 3 years. Some of them are old like ClinVar, but ClinVar keeps updating very regularly. Then there is the Genome Asia 100K project, but some of them are very new as this month. So I would recommend that you actually read these publications. It will give you in context of what is really going on, how are these genomic variations documented and analyzed, and why do you need these kind of resources when you actually have a reference genome. So you actually reference genome is just, it's like 70% one individual's readout, despite the fact that when the genome sequence was done, they were like the pooled sample of 20 individuals, but it is mostly read out from one individual. So it's not really representative of how diverse we are. And that is why you need to sequence from various populations in different contexts. Now for today, I have kept a very simple example of what we did in our lab, and I'll be very quickly describing that. So you need to focus because I'll very quickly finish this. So our lab has been interested to understand what is the role of mitochondrial DNA in human diseases. And if you look at I've just shown a representation of mitochondria. If you look at the electron transport chain, it has 5 complexes. The ones which are shown in green are encoded by nuclear DNA. The ones which are shown by red are encoded by mitochondrial DNA. It is not very difficult to make a judgment that there has to be a very intense crosstalk between the two genomes for mitochondria to function properly. And it is not just the energy metabolism. Mitochondria is involved in practically all central metabolic pathways in the cell, making it an extremely critical organelle. And given that this organelle has its own DNA, makes it a very strong case to look at what mutations in mitochondrial DNA might lead to disease phenotypes. Now, one of the things, not everybody understands the structure of mitochondrial DNA, so I have kept a very small introduction. It's a circular molecule. It is 16 kb in size. It is mostly maternally inherited. There are 2 rRNAs, 22 tRNAs, and 13 protein coding genes in mitochondrial DNA, a single non-coding region which is D-loop, and over 3,000 other proteins are required for mitochondria to function properly, which are encoded by nuclear DNA, right? And there is a very big repertoire of genome sequence data available across different populations that one can use to now look at disease association context of mitochondrial diseases. The problem with genotype-phenotype correlation in mitochondria is that when you actually see these point mutations, the symptoms manifest in many different ways, making it very difficult to understand or establish pathogenicity of a change in mitochondria. And that is where we developed an algorithm which we called as MIT-SNP score. It is published long time back. And the objective is, how do you score variation for their disease association? Again, I'm not talking of causation here, so it's very important that I keep using the word association again and again. And comparison is performed against normal data sets. So I'll very quickly show you what we did. So first things first was, I needed to understand which kind of mutations in mitochondria have an experimental evidence that this actually might cause the disease. So that was my first objective, is to first list out reported biochemical defects, which are like low complex activities, or functional studies which are done using trans mitochondrial hybrids, to establish that some of these mutations are actually causing the disease and not just associated with them. Use them as a basis to select which tools are good to now select these kind of variations from the plethora of other variation that I see. And then subsequently, I designed a scoring scheme, which I will not have time to explain today. But the whole point is, if a variant is of very little importance, it was given a score of 1. If it is of if it is really causing a problem with mitochondrial replication, say for example, it is given a score of 10. So between this 1 and 10, the other scores that you have seen here are assigned based on the anticipated impact that these mutations might have on the mitochondrial function. And then when you actually have several such mutations in an individual, we multiply all the scores and not add them. So this is the AND logic. I would request you to go and read the paper on why we applied this logic. It's very interesting. And once we did that and the pipeline was ready, we actually applied it on patient data sets, which are ataxia and MIT-SNP, and we applied it on almost 600 individuals. which are both having metabolic as well as neurodegenerative cases. Okay. For normal sequences, we use the context-dependent normal. For Indian ataxia patients, we used the Indian normal data. For Japanese disease individuals, we used Japanese centenarians so that the background variation effect is not there. And centenarians were an extremely good control set because these are people who were 100 years old and they were absolutely healthy, which means any mutation in these individuals might not be causing the disease by themselves. So when we applied this whole algorithm on the 600 plus individuals, whole genome mitochondrial DNA data sets, we could actually prioritize only 3 to 4% of total variations. So it's not 5 or 600 between any two individuals. We actually could prioritize very, very small set. And obviously, we also were able to identify novel variations in some genes which were not reported before. So it was very interesting to see that the method was working and we were able to identify some novel variants which were not even reported by that time. So basically, this is a new, this actually still is something which most math methods cannot do. One observation we made back then was that rare variants are high scoring variants as opposed to the general understanding that common variants are disease associated. We didn't observe that even at that point. And now there are more and more papers who are actually trying to scout for rare variations for disease association. So this is one of the first studies where we could actually observe this and reported it. And we could find novel disease associated variants with this application. Moving forward, again, I'll not be able to describe much of this work, but since you know that in each cell, there are several mitochondria, and within each mitochondria, there are several mitochondrial DNA, and we could actually assess this mitochondrial DNA for disease association. The second logical thing was to now look at more than 3,000 proteins which were coded by nuclear DNA but have a mitochondrial function to see the holistic impact of variation in an individual, both for nuclear and mitochondrial DNA for disease association studies. Now here we used several resources to identify which nuclear encoded proteins have mitochondrial function. And just bear with me on this slide for a minute. So if you look at the left-hand panel, we utilized several resources to identify nuclear encoded proteins with mitochondrial function, identified which of the mutations in these proteins actually have been reported to be pathogenic in ClinVar, which means they are clinically actionable, and mapped these variations onto the Genome Asia 100K project, which actually also has data on almost 600 individuals from India. We then took all these mapping where you actually found this and mapped this to DisGenNet data, which is genes and variations associated to human disease. So we could generate a mitochondrial gene disease network. And in this network, we could also identify loss of function variations from genome data sets. If some of you actually find it difficult to follow in this presentation, you can always write back to me, because there is a lot of background information one needs to know to understand this. The logic that you need to understand is what we are trying to do. We were trying to see whether pathogenic variations reported for the rest of the world, are they also pathogenic, or are they also loss of function in the Asian subcontinent, or particularly for Indian population, or not? What we actually observed is that one needs to perform population-specific mapping of clinically relevant variation. We cannot rely on loss of function variation data coming from the rest of the world to our population, because we don't see that happening. So this is very important now, that one has to generate population-specific references and population-specific variation data, which might not be reported actually for the rest of the world because they're not LOF there, but they can be here. So this is a very important message. And this recently was also endorsed by Genome AD in the recent paper, where they actually say the same thing. Now, one of the things that one can do with this kind of platform is you can study comorbidities, because now it's a network. We are saying maybe one disease, and several genes, and one gene and several diseases. So this is something that we are looking at, and particularly from Indian population context, when we did enrichment analysis, we could see that certain pathways like defects in vitamin and cofactor metabolism, tRNA amino acylation, DNA repair pathways, these are very, very important to study in Asian population context. So this is the first platform that we have identified to look at comorbidities. I didn't actually focus it from a comorbidity perspective because it requires a little bit more background to understand it at a more detailed level. But if any one of you are already working on this kind of strategy, we have actually provided all of this resource as a workflow system in Galaxy. So no programming experience is required. If you have a question, you can come on this platform and generate all your own custom workflows on the fly. You really don't have to learn anything to make this, to basically ask questions. And a lot of information is already provided as part of this platform, and we have already analyzed more than 150,000 individuals with almost 600,000 variations. So it's a very large data that we analyzed for this project. Now, some of the things that I'm not discussing today, but I would really recommend you to go and read them, is that many times you will actually hear that synonymous variations, the variation that do not change the amino acid, will not have an impact on the disease phenotype. That is not true. We actually have shown in this work that it actually can have disease association impact. And second is, since the data size that we are now looking at is going to, by 2025, in another 3 years, the genomic data will bypass the data that we generate on social media. So now you can imagine we are talking of exabytes of data. We have not even talking of petabytes now. Now that kind of data when you have, you not only have the challenge of genotype-phenotype correlation, you also have the challenge of analyzing and handling this data, and that is where we developed Frog. Again, it's not for discussion today. So basically what we are now doing with all this computational platform, you might say, what is the application of this? So the application is pretty evident. In 2016, the Mitochondrial Medicine Society actually recommended next generation sequencing as the first line of diagnosis for mitochondrial diseases. So it is not something that you might do. This is something which is a highly recommended method to diagnose mitochondrial diseases. And I'm part of the clinical domain working groups. We are actually trying to map every variation as per the approved guidelines by ACMG and AMP standards that we recommended 2 years ago and were approved by ClinGen. And now we are working as a clinical domains working group with a very large team of clinicians to see how do we actually translate this knowledge into a clinical context. Now, my lab has been developing these resources over a very long period of time, and we continue to do that from different contexts now. We are now trying to see in one of our new projects is how pathogenic variations in mitochondria make individuals more predisposed to infections. Because the other vertical of my lab work on infectious diseases, so we are now merging the two domains in a very logical context. Now, I would like you to tell that why is this important? Because these are basically the 4 tenets of personalized medicine. Personalized, as in you need to know how genetic variation drives individual treatment. It has to be predictive. One should be able to identify what kind of disease condition one is predisposed to. It should be preventive. If I know that somebody is predisposed to diabetes at the age of 30, there can be lifestyle changes introduced and the person may not manifest the disease at all. And it should be participatory, like the patient has to have informed choices and take responsibility of their own health. Now, one thing that I would recommend all of you to go back and check is the new developments happening in precision medicine. So we have evolved from the reference human genome as a reference to population-specific genomes to human pangenomes. Because now we know as and when we are sequencing more and more human genomes, there is additional 4 to 5 million sites that are getting added to the data sets, which means that we need to now capture the complexity of human pangenome with respect to precision medicine if we have to go anywhere forward from this point onwards. So have a look at the human pangenomes progress. That's something that is very, very recent and is still being discussed on how do we really do it. There are recommendations, but they are still not implemented. So this is something which is an ongoing activity. So I would recommend any one of you who is interested in genomics should be reading about this context. Now, I would like to basically take last 5 minutes to before I close is to tell you that genomics is not only about personalized medicine. And I'll give you an example here. Now, Kathleen Folbeck actually was imprisoned for killing all her 4 babies, right? It is actually said to be one of the most horrific crimes that somebody can perform. Now, the problem was that scientific evidence, so Mayo Clinic pediatric cardiologist, actually Michael Ackerman and a team of scientists, linked the death of these babies to a specific mutation which was leading to sudden infant death syndrome. So this lady actually has not killed at least 2 of her babies, is that what they could establish. For the other 2, they required data from the father, which was not available. But the whole point is that imagine genetics actually freed this woman who was convicted of killing her 4 babies. So genetics and precision medicine, this precision medicine is for health, but genetics and genomics in general have a wide array of applications. Like even within our lab, which I have not told you today, we actually use it to understand AMR. So there are a wide array of applications that you can think of. And these are all places where you can probe as future prospects for your career. Now, as I told you that my lab works on 2 domains. One is AMR and the antimicrobial resistant infections, and the other is mitochondrial diseases. I would like to show you something very different in last 2 minutes that we have done. So for some of you who does not understand what antimicrobial resistance is, so I'm actually shifting gears here. So there's no more genomics from now onwards. So I'm pretty sure all of you know about the discovery of Sir Alexander Fleming. He discovered the first natural antibiotic, penicillin. And in his Nobel Prize speech, he actually said that if we don't take penicillin or other antibiotics carefully, the bacteria will develop resistance against these antibiotics and they may no longer work. Now, this was something that he said in 1945, and this actually is coming out to be true because now we are dealing with the problem of antimicrobial resistance. It is a major issue. I wanted all of you to know about it. It is called a silent pandemic because we don't discuss it the way we discuss COVID. But WHO has declared AMR as one of the top 10 global public health threats facing humanity today. So it's a very major problem. And that is why we thought it is important to sensitize younger generation about the issue of AMR because it cannot be solved without participation from all of us. And we also wanted that this participation need not start when we are already adults. What if we can explain this concept to young children, as young as 11, 12 years old, 13 years old? Can we explain a very hard scientific concept to them in a manner which allows them to change behavior? It is not just understanding something, it should influence individuals in a manner where they are able to change their action against something which is not happening right. So I'll, with the permission of the organizers, I would like to play a 1-minute video and I hope you enjoy it. This is basically a teaser for the game so that you know what the game actually talks about. I hope it works. Here we go. So this is a video, and I thought it's important for the community, as you know, we are scientists. So if it is a problem that impacts all of us, it's something that we have to collectively do. And we am very happy that when we developed this game, it became one of the finalists in the International Educational Games competition across the world. So I would really like all of you to go to the Jigyasa or my homepage and play this game, popularize it so that we actually can contribute to this social issue, which is AMR. It's not just a scientific problem anymore. So as Mohit was saying, I work with a very, very large group of people who are not always sitting in my lab. So I do a lot of crowdsourcing projects, and I have worked with some brilliant kids. They are doing excellent work right now. These two kids happen to be my favorite because they published in international peer-reviewed journals from my lab when they were still finishing their school. So they were very, very young at that point in time, but extremely talented. And it actually, what I could see in them as talent was later endorsed by the fact that Pooja became the Gates Cambridge Fellow, Ashma became the Gold Scholar, and they both are on full scholarship for their master's and PhD programs. So this is a message to the students out there. We will be discussing more in the panel, is that you don't need really an age or a degree to do science. Passion is what should drive you for doing scientific discovery. And I have, like my experience with school kids, it's extremely rewarding in that context. A recommendation, which I'll again highlight in the panel, is that be like a Swiss knife, interdisciplinary. Today, if you are doing biology, learn at least one programming language. Very strongly recommended. The kind of data that is being generated, it will be extremely difficult if you don't know data analysis. So do that. And with that, I would like to conclude my presentation for today by thanking my lab members both in India and France. And I thank all the funding agencies who have supported our lab over the years. I would also like to thank my institute. And I'm also happy and very proud to tell you that we are among the top 4 institutes worldwide who actually contribute to biological databases, open source biological databases to the world, and they are highly cited. So this is a proud thing for India because we are actually in line with EBI and NCBI. So I'm really, really happy to share that with all of you. We do some 3D printing, not bioprinting in the lab. So if some of you are interested in doing that, you're very much welcome to come here and use this device. And of course, there are open PhD and postdoc positions in the lab. So if any one of you are interested and you saw a brief of what my lab does, if you need to more, we can chat and take it forward from there. So with that, thank you so much again for having me here, and I'm happy to take any questions.